The next important topic is Human Genome Project. The Human Genome Project was an international collaborative research program whose goal was the complete mapping and understanding of all the genes of human beings. All our genes together are known as our genome. So the main aim of this project is to understand all the genes and to complete the mapping of the genes. Mapping means finding the position of the particular gene. And what is the genome? All the genes together is known as genome. This Human Genome Project was launched in 1990 and it was completed in 2003. So it took nearly 13 years to complete the project. If you take this human genome, it is 25 times larger than the genome of any organism. And that is human beings genome was the first vertebrate genome to be completed. Approximately there are 3 billion base pairs, and the development of new area called bioinformatics is only after the completion of this Human Genome Project. And when you see about the goals and methodologies of HGP, first one is to determine the sequence and number of all the base pairs in the human genome. So what is the sequence present? To find that, and number of all the base pairs, how many base pairs are present. To find that was the main goal. And next one, to identify all the genes present in human genome. What are the genes that are present? Now we are able to find that the particular gene is responsible for hair color, then blood grouping and so on. That is only because of this Human Genome Project. Determine the functions of all the genes and identify the various genes that cause genetic disorders, and also to determine genetic proneness and immunity to various disorders also. And to store the information in databases. So whatever information we gain from that will be stored as databases, and to improve tools for data analysis, that is storing all those data in computers. Find out possibilities of transfer of technology developed during HGP to industry. So this same can be the data which is collected that can be used for industrial purpose also. The project may result in many ethical, legal and social issues which must be addressed and solved. So we have to find that issues and that has to be solved. So that these are all the goals of HGP. This project was slated to be completed for sequencing in 2003. On February 12, 2001, a formal announcement about the completion of the project was made. However, announcement of sequencing of individual chromosomes came in May 2006 with the completion of assigning nucleotide sequences to chromosome I. Now let's learn about the methodologies of HGP in detail. In the past, biological researchers were able to study only one or two genes at a time. This changed completely in 2003 with the completion of the Human Genome Project. With the entire gene sequence and the advancement in genetic engineering technology, advanced disposal, biological research could now be conducted on a large scale. For example, biological researchers could now study all the transcripts in organs, tissues or tumors. It was also possible to study how thousands of genes and proteins were together inside our cells. HGP methodology was made possible to major methodology in biological research: express sequence tags or ESTs, or DNA sequence tags and sequence annotation. In EST or DNA sequence tags, the focus was on identifying and isolating all genes that are expressed as RNA. Actually, EST is the term for all genes that expresses RNA. With EST, numerous databases of nucleotide sequences have been made available. It has facilitated the construction of preliminary transcript map of human genome. In the second methodology, the entire set of genome that comprise of all coding and non-coding sequences were indiscriminately sequenced, and later different regions of the sequence were assigned with their functions. The assigning the functions to sequence is called sequence annotation. For the purpose of sequencing, the complete DNA was extracted from a cell and divided into smaller fragments, since this is technically difficult to use a longer fragment of DNA. So these are fragments of DNA. These DNA fragments were then cloned by using suitable hosts like bacteria or yeast with the help of specialized plasmid or vectors. So with the help of DNA ligase, it is combined together and the bacterial chromosome. This process of cloning, we get many this number of copies. The vectors were called as bacterial artificial chromosomes or BAC and yeast artificial chromosomes or YAC respectively. The cloning amplified each DNA sequence, which made it easier for sequence. Sequencing was then done by automated sequencers that worked on the principle developed by Frederick Sanger, who was also responsible for directing the method of sequencing protein and nucleic acid. As per Sanger's method, the cloned DNA sequences were arranged based on their overlapping regions present. For this, overlapping segments were generated. However, arranging this was not an easy task. Therefore, specialized computer-based programs were developed and annotated the sequences and designed the chromosome. Out of the 22 autosomes and 2 allosomes, except Y, chromosome 1, that was the last sequenced in May 2006. Did you know that chromosome 1 has a large number of genes, that is 2968 genes, whereas chromosome Y has the least number of genes, only 231 genes. This challenging task would not have been possible if the advancement genetic engineering methodologies had not been put forth, that is for HGP. So this biological research was made possible only with this HGP project. When you see about the salient features of Human Genome Project, only about 5% of the genome in DNA sequence encodes proteins. So out of that, only all those genome, only 5% of that will code for or encode for proteins. The remaining are non-functional. The largest known human gene, dystrophin, it consists of 2.4 million bases. So which is the largest human gene? Dystrophin. This is a 1 mark question. The function of 50% of the genome is derived from transposable elements such as line and ALU sequence. LINE stands for long interspersed nuclear element and ALU stands for Arthrobacter luteus sequence. So 50% of the genome, it is derived from, how we are able to derive the function of the 50% of genome is by using the transposable elements, that is jumping genes such as line and ALU sequence. Chromosome 19 is the highest gene, that is the chromosome with the highest gene density, and chromosome 13 and Y chromosome has the lowest gene densities. Chromosomal organization of human genes shows diversity. So how the chromosomes are arranged, that is the human genes, chromosomal organization of human genes shows diversity, differences. 99.9% nucleotides bases are exactly the same in all people. So the remaining, that 0.1%, that is what makes the difference among a person, one person to another person. So remaining 99.9% nucleotides are exactly the same in all people. Functions for over 50% of the discovered genes are unknown. So only 50% of the function are known. The remaining 50% of the genes function are yet to be discovered. And less than 2% of the genome codes for proteins. Repeated sequences make up very large portion of the human genome. So when you see the human genome, the sequences which are found in the DNA are mostly repeated. The repetitive sequences have no direct coding functions, but they shed light on chromosome structure, dynamics and evolution, that is genetic diversity. So these repetitive sequences, they don't code for any proteins, but their function is they give they give an evidence for the structure dynamics and evolution of the particular species. Chromosome 1 has 2968 genes, while chromosome Y has 231 genes. Scientists have identified about 1.4 million locations where single base DNA differences, that is SNP, single nucleotide polymorphism, occurs in human. The single nucleotide polymorphism means the varying of one base, that is if one person, if he has the sequence A, C, T, G, the other person may have A instead of C, that will be occupied by another base. So that one difference, that is called a single nucleotide polymorphism, that occurs in humans. Identification of SNPs, that is, this is called as SNPs. Identification of SNPs is helpful in finding chromosomal locations for disease, associated sequences, and tracing human history. So that single nucleotide polymorphism helps in identifying diseases, associated sequences, and tracing the human history, that is evolution of human being. See about the applications of HGP. When you see about the applications of HGP, they are used in various fields. When you see about the molecular medicine, it is used for improved diagnosis of disease and for the early reduction of genetic predispositions to disease, then rational drug designing, gene therapy and control system for drugs, then pharmacogenomics, that is custom drugs. If you see about this pharmacogenomics, it is given in your book. It is a study of how genes affect a person's response to drugs. If you take a particular drug, the drug will not work in the same way in all persons. There will be a difference. So it is a study of how genes affect a person's response to drugs. So that is under the control of the genes. So this relatively new field combines pharmacology and genomics. That is why it is called as pharmacogenomics. Next, energy sources and environmental applications. Use microbial genomics research to create new energy sources, that is biofuels. Use microbial genomics research to develop environmental monitoring techniques to detect pollutants. So we have read about bioremediation techniques. So that is all by using this particular method. Next, microbial genomics research for safe, efficient environmental remediation. Use microbial genomics research for carbon sequestration. So how the effect of carbon can be reduced. So these all are related to the applications of HGP. In addition to this, how to manage, that is risk assessment. What are all the factors that causes risk? So risk assessment. Then in the field of bioarchaeology, anthropology, evolution and human migration. Then in the field of DNA forensics, forensic department. Then in agriculture, livestock breeding and bioprocessing. So these all together are the applications of HGP. Now we will see what are the future challenges. The information may be used for profit or for political power. So sometimes what happens is, after knowing the sequence, that it becomes easier, that is after this Human Genome Project has become very easier to determine the sequence. So some people may attempt to use this information for profit or for political power. Next one, insurance companies may refuse to insure people at genetic risk, and this would save the companies the expense of future medical bills incurred by less than perfect people. So those people, they cannot claim for their insurance. Next one, breed out to create a perfect human race. So another fear is that attempts are being made to breed out certain genes of people from the human population in order to create a perfect race. So in order to create a perfect human race, what happens is, the particular people with the genetic defect, they are breed out. Breed out means they are not allowed to mingle with the human population. That is, they will be taken, that genes will not be allowed to mingle with the other population. So to produce a healthy society, this is done. So that is also a risk of future challenges.